Alright, so we'll just wait for 2 more minutes and we'll start the session sharp at 6 p.m. If at any point you feel I'm not audible or visible, kindly raise a hand or type in the chat box. Okay, so let's start the session at 6 p.m. So this is week 4 of NPTEL live session. Good evening. Uh, So by far we have discussed about what are the different methodologies available for synthesizing a wide variety of nanomaterials, nanoparticles, and in the following session, that was week 2 of your live session, in that we discussed what are the different characterization techniques that are available to characterize the nanoparticles or nanomaterials that you have synthesized, and like we discussed a lot about different scientific instruments as well, their working principles as well, and what are the underlying theories, uh, the technical part related to those instruments and what exactly we are trying to characterize. Now, in the third session, what we discussed was the different biomedical applications that exist, or the potential of nanoparticles in different biomedical applications. We saw 3D bioprinting, we saw like how they can be used for different electrochemical and nanobiosensing, all those things. Now, uh, okay, so I think there's some mistake. It's not week 3, it's week 4 of your week 4 of experimental nanobiotechnology slide. So in this, we'll be discussing about nanotoxicology, and under this section, we'll be studying about the different in vitro methods by which we can study the antibacterial properties of the nanomaterials. Now, in the first session, you saw how to synthesize the nanoparticles, what are the different methods. In session 2, uh, you saw how to characterize. In session 3, you saw different biomedical applications. Now, in this one, we'll basically be, studying about how do we actually analyze if the nanoparticle, that is, suppose it is intended for some antibacterial action. Okay. So how would you actually verify if that particular nanomaterial or nanoparticle is actually capable of killing the bacteria or inhibiting its growth? If it is capable of the the cytotoxicity that is causing, that is leading to death of, that it is leading to cancer cell death. So basically, what are the different methods that are available? Now the third one is how to we determine the apoptotic potential. Now we'll study about this apoptosis and everything. So mostly when we are aiming for anti-cancer property. So the expectation is the nanoparticles or nanomaterials shall not cause immediate necrosis, because it would lead to a series of inflammatory responses. But in case, so we generally aim for apoptotic mediated cell death. So if your nanoparticle is capable of initiating the apoptotic mediated cell death, what are the different in vitro methods that exist by which we can actually check that? And the fourth one, in vitro hemocompatibility test, and then this one would be like continuing in the next session. So let's start. The first one is in vitro methods to study the antibacterial properties of nanomaterials. So in this we'll be discussing about in vitro methods to study antibacterial properties of nanomaterials, and which are used to evaluate how nanoparticles inhibit bacterial growth. Now there are two different terms. One is inhibit and the other one is kill. So we'll also see how these two different terms, uh, you know, can affect can actually influence or can actually decide the antibacterial properties of a nanomaterial and like kill bacteria under controlled laboratory conditions. So we'll go one by one. The different commonly used methods. We'll be discussing the different commonly used methods one by one. So the very first one that we have in this discussion is agar well diffusion method. So what we do in this method is bacterial cultures are spread uniformly on agar plates. Now you'll the following the classical method, you'll make the agar plates and you'll simply spread the bacterial culture on this agar plate overall. Like uniformly, you'll spread the agar culture uniformly like this. After that, wells are made in the agar and filled with nanomaterial suspension. So these wells will be carved out. So you can visualize it here in this particular diagram. So the wells will be carved out means this whole agar substrate, it will be carved out, it will be removed. And the wells, or I would say the, this well kind of thing that is made after carving out the agar, you will put your nanoparticle suspension in it, which is a liquid. So in this space, you will put your nanoparticle suspension. After that, you will incubate, like that would depend. Usually 24 hours is followed for incubation. You will keep it at the optimum conditions under which bacteria grows, and usually 24 hours. Clear zones around the wells called zones of inhibition indicate antibacterial activity. Now in this you'll carve out this part, suppose. Now this part would be completely like, there it will be only your nanoparticle suspension. Now if this particular nanoparticle suspension is capable of inhibiting bacteria, what it would do, it will make a zone of inhibition. Now in this part, once this nanoparticle suspension will get leached out, will be, slowly diffused through the agar, it will inhibit the growth of bacteria and you'll find something like this. This is called as zone of inhibition, which is, measured for different, like in this case, suppose you have one particular now in this case, suppose this particular example, you can see this diagram represents antimicrobial activity of silver nanoparticles, right? So we have the broad spectrum application of silver nanoparticles is checked with this experiment. So they have taken the B. subtilis, Asorius, E. coli, like both gram-positive and gram-negative bacteria, C. albicans, and this P. aeruginosa. So and from this zone of inhibition is calculated. So for different uh, you know, different bacteria, the zone of inhibition can vary. So based on that, you can determine that this particular nanoparticle or a particular nanomaterial is comparatively more effective for a particular strain than the other. So the same thing is followed here, like 100 mg per ml, 50 mg per ml, 25 microgram per ml, 12.5 microgram per ml, and 6.25 microgram per ml. So these are the, effective concentrations maybe that has been created. So agar well diffusion method at 100 microgram per ml of AgNPs and B, which is this one. So this one has been calculated for 100 microgram per ml of silver nanoparticles, right? So and B, different concentration of silver nanoparticles like from 100 to 6.25 microgram per ml with inhibition zone diameters. So this is the inhibition zone diameter. Okay, you can like pay attention to this particular diagram. So suppose this is positive control and this one is negative control. So just like any other experiment, you have to take one positive control and one negative control whenever you're doing any experiment. So similarly in this case also, so suppose at uh, the well that you carved out from this position. Okay, so you put your nanoparticle suspension here. Now, in addition to that, you'll also take one positive control and one negative control. And it's like very common. For the positive control, you'll be taking the antibacterial agent, like the known antibacterial agent that has been known for inhibiting the growth of that particular bacteria as a positive control in disc. So, since it is a positive control, so obviously it would inhibit the growth of the bacteria. So, you would see a very large inhibition zone here. But here, as you can see, media culture has a negative control in disk, right? So at this part, this is media culture, means the media in which the optimum media conditions and in which the bacteria grows. So in this case, since there is no antibiotic and nothing, no antibiotic, no antibacterial agent, so obviously the bacteria would grow and you would not observe any zone of inhibition at this point. Now this one is your nanoparticle. So like you can compare with positive and negative control, like in comparison to the antibiotic or the antibacterial agent, how effective how effective is your this I mean the zone of inhibition, how effectively it is able to control the bacteria. So I hope this method is clear. Now, moving forward, we'll see, like in this case, what we were doing, what was the sample? We were putting the nanoparticle suspension directly here. Now, another method could be this one. This is the disk diffusion method, right? Just a second. So, in this diffusion method, so same thing would be there, like in sterile filter paper discs impregnated with nanomaterial solutions are placed on agar plates inoculated with bacteria, right? So so in this case, instead of carving out the well and everything, so what you're doing is you're simply culturing bacteria. Like so, they suppose I'll draw it here. Suppose this is you cut out different circular filter paper and you impregnate your nanomaterial. Means that impregnation can you can simply dip your nanomaterials in it, or there are different methods also how you can get your nanomaterials impregnated on the sterile filter paper. Suppose you got your all your nanomaterials impregnated on this filter paper. Then the next step would be uh, solutions are placed on agar plates inoculated with bacteria. Just a second, someone wants to join. Okay, so unlike in the previous method in which the wells were carved out, in this particular method, that is disk diffusion method, what would be the procedure? So this is the inoculated agar plate. So, unlike in the previous method, right, in which the wells were carved out and then the nanoparticle suspension were poured into these wells, like this is the nanoparticle suspension, positive control and negative control. In this one, you will simply inoculate the bacteria on the in this agar plate, and after impregnating this sterile filter paper with your nanomaterial, you will simply put it here, and then you will keep it at 37 degrees Celsius in incubator, and during incubation the nanomaterials diffuse into the agar and inhibit bacterial growth around the disc. The diameter of the inhibition zone is measured to evaluate antibacterial potency. So, A and B actually are antibiotic discs, and now like with which you will compare A, B, and C. So, in this is the agar media spread on petri dish. Now, at this point, since you have simply just plated the bacteria, so this is time at time zero, right? And after doing that, you simply put these antibiotic discs. Now, maybe this could be different samples. A is different antibiotic, B is different antibiotic, and C is a different one. So, you want to compare the potential of all these three, and then simply you keep it for 24 hours, right? Now, after growth, you'll see that which one has the best potential to inhibit the bacterial growth, right? So, as you can see, the maximum zone of inhibition was observed in case of C, right? So, in rest of the plate, there will be bacterial growth, but since C had since C had the maximum potential for inhibiting the bacteria, so that is why you are observing the maximum inhibition zone of inhibition in this case. So this disk diffusion method and the agar diffusion method both are kind of similar. Now moving forward, we have the next method, that is calculating minimal inhibitory concentration, or that we call as MIC test, and the other one MBC test, that is minimum bactericidal concentration. Now, when we were talking initially, like the introductory slide, I said there's a difference between the antibacterial agent is able to inhibit, the antibacterial agent is able to inhibit the growth, and there is a difference, like both this inhibitory concentration can be different from the concentration at which the nanomaterial shows the bactericidal effect. Now we'll see how. So minimum inhibitory concentration determines the lowest concentration of nanomaterials that prevents visible bacterial growth. Okay. So suppose you added an antibacterial agent and you saw that the bacterial growth has been, reduced. Suppose this was the bacterial growth. Now after addition of antibacterial agent, you saw like the bacterial growth has been reduced. But again, it is not bactericidal at this concentration, right? Because the growth has been reduced, but the bacteria are not eradicated. Like I mean, it's not killed completely. Serial dilutions of nanomaterials are prepared in a broth, I mean the culture media, containing bacteria. After incubation, the lowest concentration showing no turbidity is considered the MIC value. So as you can see here, this is the two-fold serial dilute, the test compounds in microdilution plate, and pick a few colonies with sterile swab, prepare McFarland standard with the bacteria, and then you will simply, do the dilution. Dispense bacteria inoculate in microdilution plate with diluted test compounds, and you incubate it. And then this will be read in the plate reader, microdilution plate, to determine MIC concentration. Like at which concentration, suppose you will get something like this. So this star shows minimal inhibitory concentration. This is the positive control. This one is the positive control, and this one is the negative control, right? Now, uh, this shows no bacterial growth. So this is the dilution, matlab, means this has the highest concentration of the antibacterial agent, this has the lowest, right? Or you can consider like this as the minimal um, concentration. So as you can see, at the maximum, when you are increasing the concentration, at this concentration, whole of the bacteria is like clear. There is no turbidity, nothing that you can observe. But here, like this is the minimal inhibitory concentration at this point. Okay, so these are the minimal inhibitory. Now this could be different concentration. This could be a different concentration. Micro dilution. Suppose this is 100. So this is suppose 10. This is 0.1. So like that. I'm just explaining by giving example. Like this is how dilutions are made. Okay, so this white shows in this particular picture no growth. Similarly, like here, like there is no turbidity, no growth, and this one shows growth. So this is how you calculate the minimal inhibitory concentration. Similarly, what is minimum bactericidal concentration? So MBC determines the lowest concentration of nanomaterials that completely kills the bacteria. Means if by this, like if this is inhibition, then if you are getting a plate, you know, at this point you're not finding any bacteria or maybe just yeast. So this is considered the bactericidal effect, means it's killing all of the bacteria. It completely kills the bacteria, right? So I hope it's clear what exactly is the difference between minimal inhibitory concentration and what exactly is the difference, what exactly minimum bactericidal concentration means, along with the disk diffusion method, how we are calculating. So along with the disk diffusion method, like how we are calculating the zone of inhibition and how do we compute it with, the bactericidal effect or the antibacterial effect, and the agar-well diffusion method. Now we have so far discussed three different methods by which we can determine the antibacterial properties of our nanomaterials. Now we'll move forward to the next method, that is colony forming unit. Now mostly I personally prefer this particular method, that is colony forming unit, because it is one of the most reliable or I can say a quantifiable kind of result. Because when you're doing research, when you're in research, your data needs to be, like you can quantify your data. If you can quantify your data just and not just the qualitative analysis that you do, it becomes more significant, it becomes more reliable, and you can say that it has been experimentally validated. Now, what happens in colony forming unit count? So bacteria treated with nanomaterials are diluted and plated on agar. So what happens is bacteria that is treated with different with your nanomaterials are diluted and plated on agar. After incubation, colonies are counted to determine the number of surviving bacteria. Now this becomes important here. Suppose you start with like OD600, you recorded OD600 of zero point 0.1, right? Now you'll make dilutions. Okay, so at this dilution, like 1 is to 10 dilution, 1 is to 100, 1 is to, uh, you know, 10 to the power 3, 1 is to 10 4, 1 is to 10 to the power 5, 10 to the power 6, 10 to the power 7. Means this one is the most diluted one, right? So you'll keep on diluting the bacteria. So how I particularly do this experiment is I simply take a 96 well plate, right? And there will be different wells, right? Suppose at this point I'm only considering three wells because here it's showing three different concentrations only. So now suppose I have a bacterial suspension, just consider if it's zero point 0.1, and in this I have added my nanomaterials or nanoparticles. Suppose this is the treated bacterial suspension, right? Now what you'll do is you'll take 200 microliters from this suspension, this 200 micro, like what I do for my nanoparticles, because I work in photothermal therapy. Suppose I have done all the treatment, everything. Now I have the bacterial suspension here, the treated bacterial suspension, and now I want to see the how much viability is still there in this bacterial suspension. Now I'll simply take 200 microliters of it. Okay, now, before taking it, not 200 microliters exactly, what I'll do is I'll simply take, like you can determine the concentration, what you're taking, 10 ml or 20 ml. But before that, you'll simply first put 200 ml of, maybe bacteria or PBS in all these wells. And then after taking 10 ml, simply add this 10 ml, pipette it 2-3 times. And then after properly mixing, again take 10 ml from this well and add to this well. Okay, again mix everything. Now you'll have again 210 UL of this suspension. Again, after properly mixing, again take 10 ml from this and add this. So in this way, you'll make serial dilutions like this. You can do it differently, like separately as well, but this is how I prefer doing it. Now, this one is a different dilution. This one is a different. This one is a different dilution. Suppose dilution 1, dilution 2, and dilution 3. After doing it, I simply take 10 or 20 ml and just plate it here. I make these columns. I mean, this is not my data, but I'm just showing like how is it done. So I simply take it, I'll drop one drop, two drop, three drop, and like that. So the so this is basically done as replicates, okay? So replicates. And after that, I simply keep it at 37 degrees Celsius for the bacteria to perpetuate. And then I record what exactly is counted. So can you see the colonies? So these bacterial colonies are counted that we call as this colony forming units. So all these measurements are done like this. So I hope it's clear. Like so, when you'll take too many dilutions, right? So this one basically corresponds to this dilution. Like at this dilution, there are too many colonies which are not that you are not able to count. Because initially when you are doing the experiment, you don't know at which concentration or at which dilution, like at which concentration you are showing like what is the exact colony forming units? I mean, the bacteria with that is inhibited by the nanomaterial. So, and the middle concentration, you can find countable colonies, and then there would be some concentration also where you will not find any colonies. So this is how the serial dilutions are made, and this is how how you count colonies. So these are individual colonies that is countable. But if you see this one, it's all merged, it's all smear. You cannot count individual colonies. So that is why different dilutions are made. That is why, this is done in replicates to make it to make your data statistically significant as well. So this is one method. Now Another method by which like you can determine the antibacterial activity, like usually these are the classical methods that we discussed. This colony forming unit, determining the antibacterial activity by the MIC test, the MBC test, disk diffusion method, agar diffusion method. But nowadays, like since we know we have electron microscopy, so you can visually see if you can observe any changes in the, bacterial morphology or bacterial surface. As you can see, there are some changes or rupture maybe that you can observe. So this is E. coli, this is S. aureus, and you can see the changes in these bacteria. They are under stress. So we in our lab also we do this kind of electron microscopy based determination apart from the classical methods that are available like agar diffusion, colony forming units and everything. We also do electron microscopy of the treated bacteria in order to determine, like, in order to basically corroborate the classical findings, the findings that are obtained via classical methods. Now I would request you all to kindly at least try attempting these questions. These are some very basic questions only to, kind of revise what all five, six methods that we have just now discussed for the antibacterial activity. How do we determine the antibacterial activity? So I'll wait for maybe a minute, and I would appreciate if you can just write the answers in the chat box. And after that, we'll discuss it. Okay, okay, just 30 seconds more, and then we'll discuss the answers one by one. And in the meantime, if like while I'm discussing any topic or any method, if at any point you guys feel like there's something that you want to ask, please feel free to ask in the middle of the discussion as well. You can raise a hand or simply write your queries in the chat box also. So let's start discussing the questions one by one. So the first question reads, which method measures antibacterial activity by observing a clear zone around a well in agar? So it's very simple, obviously. Uh, this is agar well diffusion method, correct? Then the second question is the lowest concentration of nanomaterials. Now I have like multiple times stressed on this point. You have been asked a concentration of the nanomaterial that inhibits the bacterial growth. So obviously the correct answer would be minimal inhibitory concentration and not minimal bactericidal concentration. Then the next one is which method confirms whether bacteria are completely killed after MIC testing. Yeah, right. So this one, which method confirms bacteria are completely killed after MIC testing? Now let us see one by one. Now, if we look at the disk diffusion method, we know that in that we are just kind of comparing like the zone of inhibition, but the zone of inhibition will not actually give a correct, account of whether that nanomaterial is bactericidal or not. Same is with the agar well diffusion. So these two will get eliminated from the picture. Third one is gram staining. So gram staining obviously don't tell about the antibacterial effect. So the correct one is minimal bactericidal concentration test that we have just now discussed. So this will tell if the bacteria are completely killed and at which concentration you are observing the bactericidal effect. Right. So now moving forward with the next set of methods that we'll be discussing. Oh, okay. Now before that, I think we have three more questions for you because I think it's something. These are very like extremely basic questions that has been put in this particular live session only to, just kind of get it in your mind what we have just now discussed. I'll just wait for a minute maybe, and then we'll discuss. Mhm. Okay, so let's start discussing. So the first question is very simple, and the answer is there in the question itself. So in which method are filter paper discs soaked with nanomaterials placed on bacterial agar plate? So obviously the disc diffusion method. Now in the second question, let us try to understand what exactly is being asked. So in diffusion based antibacterial assay, the size of inhibition zone, suppose at this site you placed your nanomaterial, it could be like simple disc, like the filter paper that has been impregnated with nanomaterial, or maybe the nanoparticle suspension, and around it, you know, a zone of inhibition is formed. Now, this zone of inhibition basically is formed on the agar plate only, right? The here is your nanomaterial suspension. Suppose you have carved out a well on the agar plate, but on the agar plate, the inhibition zone that you see, it's on the agar plate only, right? So what it's asking, the size of the inhibition zone, the size of the inhibition zone is primarily influenced by which factor? Okay. Okay, so I think I'm basically we're discussing for this particular question, like what is the factor that actually is influenced by which factor? So this is bacterial mutation rate, diffusion rate of nanomaterials in agar, color of the nanoparticles, agar thickness only. So for this one, suppose you have put your nanomaterials here, right? Now, suppose the suspension that you have made, the property is somewhat like that it cannot diffuse in the agar only, because this inhibition zone will be visible, can be visualized, can be observed only if your nanoparticle has the ability to, at least diffuse in the agar plate. Because if it will not diffuse through the agar plate, it will not inhibit the bacterial growth. There will be complete bacterial growth all around, right? So the correct answer for this one is diffusion rate of nanomaterials in agar. Suppose you get this at 24 hours, right? At 24 hours in the incubator for the bacteria to perpetuate. But the diffusion rate, the diffusion rate of nanomaterials in the agar plate is extremely slow. Suppose in 24 hours it just travels this much distance. So obviously your inhibition zone would be very low because it has traveled at this distance only. So this is one of the limitation also of that method, the disk diffusion method or the agar diffusion method that we have just now discussed. So this is one of the limitation why CFU based methods or MIC based methods are preferred over the classical disk diffusion or the agar diffusion method. But still, you can do both. If you are trying to find out the antibacterial activity, maybe you can try proving it with a combination of two, three methods. Like you can do an agar diffusion method, then you can do the cornet forming units method, and then you can further kind of support your data through the electron microscopy images, which is like you can simply see it through your eyes. So whenever we're trying to prove that a particular nanoparticle or particular nanomaterial is having antibacterial activity, we kind of prove it via different methods. We verify it via different methods. Like if all these three different methods point towards the same data, then we can see that it has the antibacterial effect, like primary. Now, the third question reads, which limitation is commonly associated with diffusion-based antibacterial assay when testing nanomaterials? Now, I hope uh, the thing that I have discussed just now applies to this particular question as well, right? So let us read the options one by one. Right. So correct answer. This is nanomaterials will not diffuse efficiently through the agar. So this is one of the limitation. Okay, so this is one of the limitation also. That's correct because if your nanoparticle diffusion rate is too slow in the agar, obviously the zone of inhibition would be affected. So this is, one of the limitation. Now moving forward, just a second. Okay, okay, because this one is, I think this is the last set of questions that we'll be discussing for the antibacterial activity, right? So I'll just wait like 30 seconds or 40 seconds for you to at least try attempting it. Because the seventh question is, interesting. More than interesting, I would say I have like time and again repeated the difference between MIC and MBC. So I hope you guys attempt it correctly. Mm, size constraint. I didn't exactly get size. What does size constraint mean in this scenario? Okay, okay, sorry. Okay, no issues. Right. Okay, so let's attempt this question as well. So the seventh question reads, a nanomaterial shows low minimal inhibitory concentration value. It means that suppose if you have 5, 5 mg per ml, just for example, I'm telling you 5 mg per ml, 10 mg per ml, 15 mg per ml, 20 mg per ml, and 25 mg per ml. Five different concentration. Now suppose the minimal inhibitory concentration value that you found was, suppose it was uh, 10 mg per ml, right? But a significantly higher MBC value. Now, suppose the MIC value that you found with your experiments was 10, but the MBC value that you found with your experiments was 25, right? So, this is the question. This is the scenario that is being asked. Now, what does this most likely indicate? Now, based on this information, what we can kind of interpret is like the growth of the bacteria is getting inhibited at even low concentration, but still there are colonies being formed at this concentration. But this is the exact concentration when you are not finding any colonies, when there is no bacterial growth. This is the bactericidal concentration. So what does this most likely indicate? It indicates that this minimal inhibitory concentration or your nanomaterial, so it's not causing complete bactericidal effect, it's basically bacteriostatic. Okay, sorry. So when MBC is greater than MIC, the agent inhibits growth but does not efficiently kill bacteria. So that's what uh, I just said. that it can inhibit growth. This particular concentration is inhibiting growth, but it's not efficiently killing bacteria, right? So at this concentration, there are still bacterial colonies visible that can be observed. So this was the difference between MIC and MBC. Now the next question reads, which method is most appropriate for quantitatively measuring the number of viable bacteria after nanoparticle exposure? When I was discussing this method, I specifically highlighted that when you correct, right answer, correct answer. So the correct answer is CFU coroniforming units enumeration only, in which we were making different dilutions, in which like we had a plate, we divided it in different sections, and in all these different sections, basically we drop casted the treated bacterial suspension of different dilution, right? And in order to determine like at what concentration there is complete eradication of bacteria, we can like do the quantitative analysis for this particular method. Now moving forward, so just a second, I think there is some lag. Just a second. I hope it's clear. Okay, I hope it's clear. Okay, last question for this one. Why might agar diffusion methods underestimate antibacterial activity of certain nanomaterials? Now we have time and again discussed this particular point, that why do some of the antibacterial agents, if their activity is tested through agar diffusion method, they can underestimate the antibacterial activity. So obviously we have nanoparticles may not diffuse efficiently through agar because their diffusion rate is too slow. I think this one was a repetitive question, so I just straight away discussed it. So because the diffusion rate is too slow, obviously through the agar is too slow, obviously their antibacterial activity would be underestimated. There could be a possibility that it was a potent antibacterial agent, but since there was no proper diffusion, so there was no inhibition of the bacteria. Yeah, right. The correct option is C1. So poor diffusion of nanoparticles can reduce the observable inhibition zone, giving misleading results. Right. Now we'll be talking about in vitro cytotoxicity analysis. In our previous live sessions, we have talked about different uh, anti-cancer like the nanoparticles that has been being used for anti-cancer effects, or for different, basically a lot of uh, cancer, against a lot of cancer. So there are different methods, there are different in vitro cytotoxicity methods by which we can analyze the percentage of live and dead cells in that matter. Now, when we talk about in vitro cytotoxicity analysis, this is a kind of an umbrella term. It would encompass a lot of different methods. Now, these are the basic underlying principle on which these particular methods actually work. We'll deal it one by one. We'll understand. Just a second. So we'll one by one pay attention to what these methods mean and what are the underlying principle on which these methods are based. So the very first one is dye exclusion method. So the principle is dead cells allow dyes to enter. Now why does it happen? Because dead cells have compromised cell membrane, right? But live cells have intact cell membranes, intact plasma membrane, right? So this is live, this is dead. Now, what happens with dead cells? Because their membrane integrity is compromised, the dyes can easily get in through the cells. So when you will see it under the microscope, suppose you added some dye, if I'm talking about this dye exclusion method. So since the dead cell have compromised membrane integrity, the dye can easily penetrate, but the dye cannot penetrate in live viable cells because they have uh, perfectly fine membrane. Now, since it can penetrate here, so these would appear colored under the microscope, but this one would not appear colored. So this is the basic principle. So dead cells allow dyes to enter while living cells exclude them. That is why it's called as dye exclusion method, dye exclusion assays. Method cells are mixed with a dye and observed under a microscope. So the very common one is trypan blue exclusion assay. Live cells remain, just now that I have told, live cells remain colorless, but dead cells appear blue in trypan blue case. Now there are other methods also that we'll go one by one. Now the second assay is colorimetric based metabolic assay. So living cells convert certain substrates into colored products through metabolic activity. So this happens through the internal metabolic activity of the cells. You will give some material, it will go inside the cell and it will be converted into some different metabolic product that you will be detecting colorimetrically. So measurement, absorbance using a spectrophotometer, we'll discuss about this MTT, XTT, all this, how it happens. The other one, this one is the colorimetric assay. We are recording absorbance. Based on absorbance, we are seeing, because suppose uh, in some samples the absorbance is too high, means the metabolic product that is produced by the viable cells are too high. But in case of dead cells, the metabolic products might be too less. So I'm just giving one example. Now the other one is fluorimetric based methods. So in this we have Alamer blue and CFDA, fluorine diacetate, acetyl methoxy assay. So fluorescent viability assays. So in this case, basically you determine by a difference in fluorescence. So fluorescent dyes distinguish live and dead cells based on membrane integrity and enzyme activity. And viable cells convert non-fluorescent substrate into fluorescent products. If you remember what we discussed in the earlier sessions. So this calcine AM about which we talked about, so this is generally non-fluorescent. Now, when we when it is taken up by the viable cells or the live cells, it gets converted to like this. This acetyl methoxy group is removed, and only calcine is left behind that gives green fluorescence, right? So this fluorescein diacetate AM assay is this one. So this is based on the fluorescence, the difference in fluorescence. Like this one is non-fluorescent, but this one is fluorescent. And the last uh, approach is measure light emission produced by biochemical reactions. So there are certain biochemical reactions that happens inside cells. Like specifically, if I talk about linked to cellular ATP or metabolic activity. So higher luminescence indicates that number of metabolically active cells are also higher. Now we'll see a little. Uh, we'll talk about these methods in a little briefly. Now let's talk first about this trypan blue assay or trypan blue exclusion principle, because this one is like very commonly used. So trypan blue stain is a dye used to determine cell viability. It works on the dye exclusion principle where live cells exclude the dye while dead cells absorb it under a microscope. Now it will look like this. So these are the dead cells. The blue ones that you are seeing, these are the dead cells, and these are the live cells. So whenever we are doing any experiment and we are suppose we said that we did experiment on 10,000 live cells, or the seeding density was 10,000 cells or 12,000 cells per well, or like that. So we have, how do we do the cell counting? So I'll just very briefly, very quickly explain how do we do that. Suppose you cultured cells in a disk, and after reaching, once these cells reached 80% confluent, means this is the growing flowers, and once it reached 80% confluent, means it is the cells have covered almost 80% of the area. What you do is the old medium is removed, the cells are washed, and then you add trypsin. Like if these are adherent lines, so you basically add trypsin in order to detach these cells that are attached on the disc. Now once the cells are attached, you make this single cell suspension, means you mix it, uh, then you'll centrifuge it to get a cell pellet. Now once you get the cell pellet, you'll again, suppose this is the cell pellet, right? Now you'll again resuspend this cell pellet in fresh media. Now you'll take 10, what you do is you take 10 ml of this, and then of this cell suspension, and to this you add 10 ml of trypan blue dye. Mix it well and then add it here in this hemocytometer. So for those who don't know what exactly is hemocytometer, it is a small device, I would say device that looks like this. Now, this area that you are seeing here, if you see it under the microscope, it will look somewhat like this. It will look like this, right? You'll see these columns. Now, what you do is with the help of pipette, you just simply add 20 ml. You'll charge the device, you'll add 20 microliter of the sample, which contains like 10 ml of your suspension and 10 ml of your the dye, the trypan blue. You'll add it from this place, and it will go and simply, once you have added it from here, it will somewhat look like this, like this. So those cells that would take up the dye are the dead cells, and then those who do not take up the dye that appears clear, colorless are actually live cells. So you have to count the live cells, and then there are calculations based on which you exactly calculate that if you want to see 10,000 cells, how much cell suspension you need to take and mix. So this is what like trypan blue exclusion, trypan blue staining works. It's something that is routinely followed whenever doing cell culture and you are seeding cells. It is one of the most followed approaches for cell counting also. So I have told this one, this is the alive cell. You can see the membrane is not compromised, but in dead cells, the membrane is compromised. That is why trypan blue enters the cells and stains the cell blue. So this is hemocytometer. This trypan blue staining is routinely followed for this hemocytometer based cell counting. I hope it's clear. We'll just read it once. It works on the dye exclusion principle where live cells exclude the dye while dead cells absorb it. Under a microscope, live cells appear clear and dead cells appear blue. And it is commonly used for cell counting and viability assessment using a hemocytometer. Now moving forward to the next method is Congo red staining, which is more or less same to the previous one that we have just now discussed. So it is a dye-based method used in vitro cytotoxicity assessment to study cell morphology and detect cell damage or death, as you can see it here in the images. Sorry. Congo red binds to cellular proteins and extracellular materials. Damaged or dead cells take up more dye. Now this is important. Cells are treated with the test compound, fixed, stained with Congo red, washed and observed under a microscope. Increased staining intensity or morphological changes indicate cell damage or cytotoxic effects. Now, they would be in the dead and damaged cells will take up more dye and they will appear, you say, more intense colored. Used in cell culture studies to evaluate drug toxicity, nanoparticle toxicity, and biomaterial biocompatibility. The next one is erythrocyan B assay. So mostly the trypan blue one is majorly followed, majorly used, but since it's there, it's one of the methods. So we'll be discussing this one as well. So erythrocyan B is a fast, inexpensive, like comparatively cheap and less toxic dye used in cell viability assays to distinguish between live and dead cells. It acts as a substitute for trypan blue. Substitute for trypan blue, staining dead cell membranes while leaving live cells clear, and is effective for various cell types including yeast and bacteria. Now, this one is very effective for yeast and bacteria as well, right? So, advantages, what are the advantages over trypan blue? What are the different advantages of erythrocyan B over trypan blue? That it is less harmful to cells and it often allows for more accurate viability counts. Stability, stable under varied conditions, reducing artifacts in measurements. But trypan blue is generally preferred over erythrocyan B for cell viability assay due to better reliability and ease of observation. Now, if you'll see here, so like it's not very easy to observe and count the live and dead cells. But in case of trypan blue, it was like clear cut. You could see simply. Dead cells stain deep blue while live cells remain colorless, high accuracy, and the background interference is less in case of trypan blue. And that is why it has more wider use in comparison to erythrocyte B. Moving forward and coming to the colorimetric assay. Now, so far, what we were discussing about the dye exclusion principle, where we were determining the live and dead cells based on the dead cells would take up the dye and the live cell won't take up the dye. Now, coming to the colorimetric based assay, the very first that is like commonly used, that is most widely used, is the MTT assay. Now, what exactly is the MTT assay? Let's understand that. So, the MTT assay is a colorimetric assay used to measure cell viability and cytotoxicity based on the metabolic activity of living cells. So, this one is based on the metabolic activity. The previous one that we were talking about, it was basically like if the cell membrane is, compromised or damaged, the dye would penetrate. Now, this one is based on the metabolic activity, because live cells, the metabolic activity of live cells would be different from that of a dead cell. Now it is based on this particular principle. It uses a yellow tetrazolium salt. It uses a yellow tetrazolium salt that you see here. So metabolically active cells, viable cells or live cells contain mitochondrial enzymes such as succinate hydrogenase, right? So the live cell or viable cell will have mitochondria which will have succinate dehydrogenase enzyme. What do this? What does this succinate dehydrogenase enzyme does? These enzymes reduce MTT, this yellow tetrazolium salt that you are adding. Suppose you took a 96 well plate. Now understand this. I have made this schematic. Suppose you have a nanomaterial suspension, nanoparticle suspension, right? Now you want to check at which concentration, you are observing the maximum cell toxicity, or maybe you want to see the concentration at which at least 50% of the cells are killed. Now suppose this is the plate, you do it in triplicates, okay? And this is on coming down, increase in concentration. Suppose this one is control, and then 5, 10, and 15, 20, 25, 30, 35. Suppose this one is 35. Suppose 35 microgram per ml. So increasing concentration, with increasing concentration of nanomaterials, you treated the cells. So this diagram is made only to make you guys better understand. Now this nanoparticle suspension, different dilution of this nanoparticle suspension is added to the cells that is already grown in this 96 well plate. Now, the next step would be, once you have, suppose you have followed a 24-hour incubation, you incubated the cultured cells with your nanoparticle of different concentration for 24 hours. After 24 hours, what you'll do is you'll discard the spent media, or maybe the usual protocol is just wash the cells at least twice thrice with PBS, and after that add the working suspension of this yellow tetrazolium salt, this add this MTT, and after adding MTT just incubate it for 3 hours at 37 degrees Celsius, right? After incubating it for 3 hours, take the plate out, discard all of it, and add the solubilizing agent, that is DMSO. Now, why is it happening? Why are we adding DMSO? Let's read it. So these enzymes reduce MTT, this yellow tetrasodium salt that you have added. So this succinate dehydrogenase enzyme will reduce this yellow tetrazolium salt to insoluble. This is insoluble formazine crystals. Now this insoluble formazine crystals needs to be solubilized. So in order to solubilize this formazine crystals, we add DMSO. Like this is a common chemical used. These enzymes reduce MTT into insoluble purple crystals called formazine. Dead or damaged cells cannot reduce MTT. Now, dead or damaged cells will not have an active mitochondria, it will not have succinate dehydrogenase enzyme. So, it will not be able to produce these insoluble purple crystals, which will later be solubilized. And once you add DMSO, the solid crystals that has formed, it will stabilize and give purple color suspension, purple color solution that you see here. So suppose if we see this particular plate, this particular experimental plate, so suppose this is control, and in this way you have added the nanomaterial, increasing concentration of your nanoparticle that you have added. So you can see like from this concentration, if you compare with this one, there is decrease in production of this purple solution, which indicates that the cells are getting dead, because dead cells cannot produce this insoluble purple formosine crystals, which can later be solubilized. Now you'll take this plate, put it in plate reader, and record absorbance at 570 nanometer. And based on the absorbance, you will generate the data, which will somewhat look like this. So you have this plate, put it in the plate reader, right? Now these are different concentration. Now based on this graph that you will get, percentage cell viability, you will plot it, you'll find this IC50 value. Because like whenever you are submitting a research paper, when you're talking about any nanomaterial, you will be asked, what is the IC50 value of your nanoparticle? This IC50 value is basically the concentration of nanoparticle at which 50% of the cells are dead. So this IC50, half maximal inhibitory concentration, is the concentration of a substance. It could be a drug, it could be a compound, it could be a nanoparticle, plant extract, or anything that you are using for the uh, you know cytotoxic effect, required to inhibit 50% of cell viability or biological activity in a cytotoxicity assay. So this is how you do the IC50 calculation. Now moving forward to the next method is MTS assay. So this MTS assay is very similar to the MTT assay that we have just now discussed, except one step, which was the solubilization step. So in MTT, we were adding DMSO to solubilize. Suppose these are the purple formosine crystals. Now if we'll simply put it in plate reader, it can this since these are solidified crystals, so you'll not be able to get the clear absorbance recorded. So that is why it is diluted, it is solubilized by addition of DMSO, so that this is the solution and you can record the absorbance. Now in this step, the MTS is a colorimetric method used to measure cell viability and proliferation based on metabolic activity of living cells. The reagent MTS is reduced by cellular enzymes in metabolically active cells, similarly to what we have seen in MTT assay. In MTT assay, there was succinate dehydrogenase enzyme that was reducing the yellow tetrazolium salt to formosine crystals. Now, in MTS assay also, there are cellular enzymes uh, present in the metabolically active cells that reduce the MTS, this MTS compound, MTS tetrazolium salt reduction, which leads to colored formosine dye. Now, this colored formosine dye that forms from this particular MTS tetrazolium salt, you don't need to there's no solubilization step. So this reduction already produces a water soluble colored formosine product. So the amount of colored formed is proportional to the number of viable cells, and in this case, in previous case, we were recording absorbance at 570 nanometer because the salt was different. In this case, the absorbance is recorded at 490 nanometer using a spectrophotometer. So this formosine product is water soluble, so no solubilization step is required. This is one of the major advantage of MTS over MTT assay. Simple and faster than the MTT assay. So obviously there is no solubilization step, so it's comparatively faster. Now moving to the next method of determining in vitro. So like in this slide, I have simply mentioned that in this one, in MTS, okay, so this is MTS only. So in MTS, these groups they contain charge. Now the question is, like I have just now told, that there is no need for solubilization. Now the question is, why there is no need for the solubilization step? So the reason is, this MTS reagent that is being used to treat the cells, it contains charged sulfonate groups, right? It contains charged sulfonate groups in its molecular structure that is very well visible here, right? These groups make the molecules more polar and hydrophilic. This particular charged sulfonate groups makes this MTS molecule more polar and hydrophilic. And when it is reduced, it forms formosine that remains soluble in the aqueous culture medium. So this is the reason, right? And in addition, in comparison to this, the MTT forms insoluble purple formation crystal inside cells. That is why it needs to be solubilized using either DMSO or isopropanol. So majorly DMSO is used. Now the next one is XTT assay. So again, it is also a colorimetric assay used to determine cell viability, proliferation, and cytotoxicity. And the reagent XTT is reduced by mitochondrial enzymes in living cells. It forms an orange colored water soluble formation dye. So in this case also water soluble formation dye is formed, but it is orange in color. In MTT it was purple colored. So obviously since there is change in the color, obviously the absorption uh, maximum absorption wavelength would also differ. So the color intensity is measured at 450 to 500 nanometer, and the advantages is it's more sensitive, water soluble, and suitable for high throughput streaming. So most of the colorimetric methods, the basic idea, the basic underlying principle is same for all these methods. It's just slightly different. And now suppose, now one more important aspect to this scenario is, suppose the selection, like which method to select. Suppose you want to decide that for your particular nanoparticle that you have made, I, like I often draw this diagram. This is basically MCT, microcentrifuge tube. Now you have your nanomaterial in it. Now suppose you want to decide that for your nanoparticle, which method would be best to use. Now, usually when you make nanoparticle, you also record its absorption spectra. Now, suppose its absorption spectra lies somewhere in, in the region, like suppose up to 500 nanometer. It shows intense absorption. Now, if it is used in low concentration, it's fine. But if it's used in high concentration, this absorption can interfere with the absorption you see for the this XCT for this reagent. Now, so in that case, what you can do is, like you can do MTT assay for this one, because in this you will be seeing at 570 nanometers. So like it differs. There are different assays available. You can and if in that case also your the absorption of your compound is not aligning with any of the colorimetric methods, you can switch to fluorometric methods. There are different fluorometric methods also. Like if the absorption is not aligning with the commonly available these colorimetric assays, XTT, MTT, MTS, there are different fluorometric methods as well. Like we'll see the Almer blue assay, etc., that you can use for fluorometric based detection. So the next one is sulforhodamine B assay. So this sulforhodamine B assay or SRB assay, it measures cell density based on total cellular protein content. So it binds to basic amino acid residues of cellular proteins under acidic conditions, and after staining, excess dye is washed away and the bound dye is solubilized. The intensity of color is measured spectrophotometrically. So absorbance is taken at 540 to 565 nanometer, and used to determine cell growth, proliferation, cytotoxicity. So these are different methods, different colorimetric methods: MTT, MTS, sulforhodamine B, XTT. The next one is crystal violet assay. Now this is a little different. So in the crystal violet assay, what you do is it works only for adherent cells. So as you know, when we are culturing cells, cells are either adherent, that grows while being attached to the disc or flask in which you are growing, and the other one is suspension cells. Like they do not get attached at the surface of the flask in which it is being grown. So the principle is, this assay measures cell viability based on staining of adherent cells. The dye crystal violet binds to DNA and proteins in intact cells. After staining, dead cells that detach during washing are removed. The dye retained by adherent viable cells is solubilized and quantified, and measurement absorbance is measured at 570 to 590 nanometer. So it is now one of the limitation of crystal violet assay is it works only for adherent cells. And one of the limitation again is, sometimes during the process when you are washing, when you are adding dye, when you are handling it, some of the like healthy or live cells can also get detached in this process. So dead cells that detach during washing will not be counted. So you'll not have not ideal for measuring metabolic activity, unlike MTT assay, which measures the metabolic activity. Now we'll talk about the fluorometric based assay. So far, we have seen the dye exclusion principle, dye exclusion based methods. Next, we saw the colorimetric based uh, methods, colorimetric based in vitro methods. Now, after colorimetric based, like the absorbance based, basically, we'll be seeing the fluorometric based methods. So, in this, this is the most commonly used one, that is Hallamar blue assay. Just a second, I just want to drink water. Okay, I'll just check. Okay. Coming back to our topic, Alamar Blue assay. So the Alamar Blue assay is a non-toxic, highly sensitive, fast colorimetric fluorometric cell viability reagent used to measure metabolic activity. Now this Alamar Blue assay, again, it measures the metabolic activity. It utilizes the redox indicator dye resazurin, that is blue and non-fluorescent. So initially, this Alamar Blue dye, the compound is resazurin. It is blue in color and it has low fluorescence. Okay. Now, once it enters the viable cell, so these live cells or these viable cells, what does it do? It reduces this resazurin, okay, oxidase Alamar blue reagent, and it is non-fluorescent. It gets reduced Alamar blue reagent, which is fluorescent red. So when it gets reduced, so it leads to formation of resorufin, that is pink, and it has high fluorescence. So low fluorescence, high fluorescence. This is what is detected in this. So let's read how it happens. So it is a cell viability assay used in vitro cytotoxicity studies. It is based on the reduction of resazurin blue non-fluorescent to resorufin pink fluorescent by metabolically active cells. So only metabolically active cells are capable of doing that. So suppose these are different cell suspension. This one has most number of live cells, right? Suppose this one has fewer number of, and this one has the least number of live cells. So what do you expect if you are doing the Salamar blue assay? Since this one has most number of live cells, it will convert most of the resazurin added to resorufin, which is pink in color and fluorescent. So the fluorescence intensity would be highest in this one, a little low in this one, and least in this one, right? So based on this, you can compute your data. So this is the basic principle on which it works. So the principle is living cells convert resazurin to resorufin through cellular metabolism. The procedure is add Alamar blue reagent to cultured cells, incubate for a few hours, then measure color of fluorescence using a plate reader. High fluorescence means more viable cells. I have explained it just now. Advantages: non-toxic, sensitive, and allows real-time monitoring of cell viability without killing cells. Now, this is very important. Now, this one I have already explained, this fluorescein diacetate AM. So, this fluorescein diacetate FDA hydrolysis is a biochemical method used to measure the enzymatic activity of living microbial populations, often serving as a proxy for total microbial activity. Like it is written in generalized way, like for soil cell viability. This is a non-fluorescent estrase substrate. Now, when this enters the viable cell, these viable cells have cellular esterase enzymes, which removes this uh, AM group. This AM group is removed, and once it is removed, it now this is non-fluorescent. But once this group is removed, it becomes fluorescent. It gives green fluorescence. So this is a fluorometric method that is used for cell viability. Typically, it uses an excitation wavelength of 488 to 490 nanometer, and the emission wavelength is recorded from 513 to 530 nanometer. Now, so far, what all four methods we have discussed? We have discussed the dye exclusion method, trypan blue, Congo red, all these things. Then we discussed the colorimetric methods, in which we talked about MTT assay, MTS assay, XTT assay, and then I think, okay, these are some like some of the examples. Colorimetric based assay. Then we talked about fluorometric assay, especially the Alamar blue one, which is most commonly used. Now, after these four, colorimetric, fluorometric, dye exclusion, the another method is based on this luminescence that is recorded. So this is luminometric ATP assay, which is one of the luminometric methods. So what happens in luminometric ATP assay? It measures cell viability. How? By detecting intracellular ATP levels. Now live cells will obviously have ATP production for it to sustain, so which reflect metabolically active cells. It uses luciferin-luciferase reaction where ATP produces light, and once the light is produced, this light is detected, the luminescence is detected, and suppose if the luminance is high, means the number of viable cells is also high. If the luminescence is less, means the number of live cells are also less. So again, it is based on directly proportional to luminescence. So how does this happen? The cells are seeded in 96-well plate and incubated overnight to allow attachment. After that, once the cells are confluent, mostly like confluent in the 96-well plate, we treat it with the test compound or drug or nanoparticle and incubate for the required time, like sometimes 24 hours, 48 hours, 72 hours, based on the after like how long it takes for your drug or nanoparticle to do the action. And then what is done? Add ATP detection reagent. So add the reagent containing luciferase and luciferin, which lyses the cells and reacts with intracellular ATP. So then incubate for 5-10 minutes at room temperature so that this luminescence can occur, this luminescence reaction can occur, so that you can finally record the luminescence using a luminometer or plate reader. So this is the chemical reaction that is happening. And the other is real-time viability assay. Again, this is based on luminescence only. So you can have a track, you can have a track of how this luminescence is changing. Based on this luminescence, you can see how the percentage cell viability is increasing. The process is more or less like same that we have studied. Now try attempting this these questions. I think I have written an answer of one. Maybe I have not put animation on it, but let's try attempting these questions. I would really appreciate if you guys uh, try attempting it. I will wait for a minute or two, maybe. These are simply like straight away you can answer it, right? Okay, so let's discuss one by one. So the first one is which assay determines cell viability based on mitochondrial metabolic activity converting tetrazolium salt into formation crystals. Very simple one, the MTT assay. The second one is which assay is commonly used to evaluate long-term survival and proliferative ability. Now this one is something I think I have discussed the crystal violet and all those things, but I have not mentioned this term. So this is colonigenic assay. Sorry, this is colonigenic assay. So what is done basically when we talk about long-term survival and proliferative ability. So for this, suppose we have already treated the bacteria and everything. Now we add colonies, and it is applicable both for bacteria and for cells also. And then we see if after treatment and everything at what dilution it is able to form the colonies. If you can see the colonies, because even after treatment, even after everything, it is still like from it is able to form colonies from a single, from single colony, it is able to proliferate, means the therapeutic efficacy was not that effective. So for long-term survival and proliferative activity, this colonogenic assay or colony forming assay is followed. Third one is which assay is commonly used to evaluate. I think I have added this question twice by mistake. Okay, singles. Yes, I think I have added it. I'm so sorry. It's okay. Uh, this has been repeated twice. My bad. Sorry for it. It measures the ability of a single surviving cell to form a colony over time. So we see that if this single surviving cell has the ability to further form a colony. Now the next section is what are the different in vitro methods to study the apoptotic potential of nanomaterials. Now I have already discussed apoptosis, I have discussed necrosis, and I have also discussed why nanomaterials are intended for apoptotic mediated deaths and why not uh, immediate necrosis. So, in this, we'll be discussing about in vitro methods to study the apoptotic potential of nanomaterials, in which they are used to determine whether nanomaterial induces programmed cell death, apoptosis. Okay, we want programmed cell death and not immediate necrosis because this could lead to inflammation and other and cascade of immune responses that isn't good. In cultured cells, common techniques detect morphological changes, DNA fragmentation, and some different apoptotic markers are also there uh, that comes into the picture when during apoptosis, which can be detected by different methods. Now let us see those methods one by one. So the very first one is annexin V assay. So what happens in annexin V assay is this method detects phosphatidylserine exposure on the outer cell membrane, an early sign of apoptosis. So, during apoptosis, what happens is now suppose this is normal cell membrane and there is no phosphatidylserine moiety on surface, right? This is normal cell. There is no phosphatidylserine moiety on surface. Now, apoptotic cell membrane. This is apoptotic cell. This is normal cell membrane. This is apoptotic cell membrane. Now you can see a phosphatidylserine moiety on the surface, right? So apoptotic or necrotic cell membrane have phosphatidylserine on surface and this membrane over the time disintegrates. So this is what happens in case of apoptosis. And cells are stained with annexin V, often with propydium iodide and analyzed by flow cytometry or fluorescence microscopy. So this is healthy cell. This is early apoptotic cells. Okay. So this phosphatidylserine moiety that is present like here on the inside in normal cell membrane, what happens during early apoptosis, this phosphatidylserine moiety, it flips out. It flips out. Okay. So this is the difference. Phosphatidylserine confined to the inner leaflet. It is confined to the inner leaflet. Cell membrane is intact, luminescence is negative, fluorescence is negative. Now, in case of when apoptosis sets in, this phosphatidylserine translocation to the outer leaflet, it comes here and it flips out. Cell membrane intact, luminescence positive, fluorescence negative. Now, this secondary necrosis, Like this is just an explanation what is happening. PS inner outer leaflet. Fourth, cell membrane compromised. Luminescence positive. Fluorescence is also. This is like kind of discussing a method, but in this case, this is a programmed cell death. Then after apoptosis, obviously there would like it would continue. Then it will lead to complete necrosis. Just a second. Now, the second after annexing V assay, the second one is the tunnel assay. So what does it? What happens in this? The DNA strand breaks generated during apoptosis. So there are this tunnel assay basically detects what are the different DNA strand breaks happening or being generated during apoptosis. So the enzyme terminal deoxynucleotidal transferase. This is the enzyme. It labels the fragmented DNA ends with fluorescent nucleotides. This it is being labeled with the fluorescent nucleotides. This enzyme is making it possible. Now allowing apoptotic cells to be visualized using fluorescent microscopy or flow cytometry. So this is the procedure. Cultured cells are exposed to the test nanomaterials for a specific time. Like simply you will take the cultured cells and you'll incubate it with your nanomaterials or nanoparticles to induce apoptosis, like to induce cell death. Now, after the required time of experiment 24 hours, 48 hours, 72 hours, cells are fixed using the classical method, paraformaldehyde fixing, and then permeabilized with a detergent. Now, this permeabilization step are sometimes carried so that the dye or the fluorescent agent with which you are treating it, it can actually penetrate. So in order to allow reagents to enter the nucleus, that is why at a very low concentration, Tritonex is handled, which makes the membrane partially permeable. So that is why this Tritonex 100 is added to allow reagents to enter the nucleus. Now comes the tunnel reaction part. The enzyme terminal deoxynucleotide transferase adds labeled DUTP nucleotides to the free OH ends of the fragmented DNA. Now, if there will be apoptosis, there will be this fragmented DNA present present in apoptotic cells. Cells are incubated with the tunnel reaction mixture for 30 to 60 minutes at 37 degrees Celsius, after which it is the data or the results are read either through fluorescence microscopy or through flow cytometry, like FAGS based analysis. SIM is being shown here, wash and then fluorescently labeled DNA fragment. Suppose if the DNA break has happened here, it will come and bind it here, and you can simply, see the image. Now, what before coming or before going to the next method that is based on some of the apoptotic markers or what is uh, in the cascade of reaction that follows once the apoptosis sets in, how do we detect that? So, apoptosis is a form of programmed cell death orchestrated by a proteolytic cascade of cysteine aspartic proteases. Do remember the name, or you can also remember the short form, that is caspases. So whenever there will be talk about apoptosis, there will be caspases coming into the picture. So initiator caspases 8, 9 and 10 are activated via dimerization complexes. Example, apoptosome. So the usual mechanism is once the membrane integrity is compromised or whenever the cellular homeostasis is disturbed, it leads to production of cytochrome C, okay, which binds to APAF1 to form apoptosome, which will lead to release of caspase 9, which then cleave and activate executioner caspases, resulting in cellular dismantling, membrane blebbing, and DNA fragmentation. So this is the mechanism. Like this stress, like this is the intrinsic pathway of apoptosis. This is extrinsic pathway. This is death ligand and death receptor, and this is the other method. Usually, uh, like most of the nanomaterials usually follow this intrinsic pathway. There is DNA damage, cell stress, changes in mitochondria. So what happens is whenever there is damage to mitochondria, okay, the electron transport system is disturbed, which leads to leakage of the electrons, and which further, binds to the oxygen, leads to production of reactive oxygen species. Now, this reactive oxygen species also aids in production of, like, also aids this apoptosis and caspase production and everything. So these are the different pathways, but ultimate is cell disassembly, membrane blebbing, and DNA fragmentation. Now, membrane blebbing also is one part. I'll show one of my data. Like it's kind of very interesting. We have seen the blebbing on cancer cells. Suppose this is cancer cells. We have seen this membrane blebbing via scanning electron microscopy on these cells. It's not published actually yet, so that's why I'm not showing it here because it will be in the public domain. But like if your nanomaterial is actually inducing apoptotic mediated cell death, it's following up that pattern, you will see the membrane blebbing. Now, since the reason why I was talking about caspases, what exactly are caspases when they are released, is because we were supposed to study this particular method, caspase activity assay. So, apoptosis involves activation of caspase enzymes we have just now seen in the preceding slide, particularly caspase 3, caspase 8, and caspase 9. These assays measure the enzymatic activity of caspases using colorimetric or fluorometric substrates, indicating whether nanomaterials trigger apoptotic signaling pathways. Now, so this is the schematic. Suppose uh, this is the colorimetric probe activator. This is colorless, and let's understand the procedure. When we'll read this, we'll kind of understand what the schematic means. So, cultured cells in a plate and treat them with a nanomaterial or test compound for a specific time to induce apoptosis. Now, after incubation, cells are harvested and lysed using a lysis buffer to release intracellular proteins, because whatever intracellular proteins, these caspases and everything are produced, it is happening inside the cells. So you need to lyse the cells in order to get the intracellular proteins, which will also include caspases. Now, suppose this is the viable cell. Okay, you lysed it and got all the cellular proteins. Now, this cellular protein will also have caspases, right? Now, add a specific substrate that can be cleaved by activated caspase. Now, this cellular protein that you have collected, it will have caspases. Now, if we add some substrate that can be cleaved by caspase, right? So, it will lead to decrease in the amount of that substrate that you have added. Now, you know the initial amount of this substrate that you are adding, and with different dilution, you can see that in some fraction the decrease in amount is less, in some other fraction it is more. So incubate the reaction mixture for about 30 to 60 minutes at 37 degrees Celsius to allow substrate cleavage. Measure the colorimetric or fluorometric signal using a microplate reader. So increased signal indicates higher caspase activity and apoptosis. Now suppose this is the probe that you are adding, this is the substrate, suppose this is the fluorescent tag substrate. Now your cellular proteins from the lysed cells will have caspase 3. This caspase 3 will cleave this substrate, and it will lead to the fluorescence emission. There will be different in the fluorescence emission that you will observe, that you can record in plate reader. Now this is caspase activity test. You need to know what is apoptosis. You need to know how these executioner and all these caspases are produced intracellularly, and what basically are the you know stress responses under which triggers this apoptosis. So you need to know all of it. Now the other method to determine this apoptosis and everything is the DNA fragmentation assay. Because obviously if there is cell dismantling, there is disturbance in cellular homeostasis, there is DNA damage happening. So in that case, during apoptosis, cellular DNA is cleaved into internucleosomal fragments. When analyzed by agarose gel electrophoresis, these fragments produce a characteristic DNA ladder pattern, right? It will produce a characteristic DNA ladder pattern. Now, I think this row, sorry, this column, like it doesn't have caspase 3, this one has caspase 3. So, this DNA has been cleaved and there's a different kind of, ladder pattern that is produced. Same thing is, sorry, there is DNA cleavage at this point, at this point, at this point. So there is Once you do the DNA electrophoresis, you'll find a characteristic ladder pattern with which you can determine difference between intact DNA, DNA ladder, intact DNA, and the cleaved DNA, right? So this is again one of the method by which you can kind of validate or kind of verify that okay, the apoptosis has happened, DNA fragmentation has happened. Now, before discussing the next method, let's understand what exactly is mitochondrial membrane potential and how this mitochondrial membrane potential gets disturbed during this apoptosis. Just give me a second. Okay. So, loss of mitochondrial membrane proteins, loss of mitochondrial membrane potential is a key event in apoptosis. Fluorescent dyes such as JC1 are used to monitor mitochondrial integrity. A decrease in fluorescence indicates mitochondrial dysfunction caused by nanomaterial exposure. Now, don't pay attention right now to the JC1 staining and everything because we'll see in the subsequent slide. Just focus on how, like in cell that has perfectly fine cellular homeostasis, this mitochondrial membrane potential is maintained, as you can see here, like the continuously working electron transport chain and everything. Now, once there is mitochondrial damage in response to cellular stress, or whenever there is a disturbance in the cellular homeostasis following apoptosis or cell death, so this membrane potential that you see here, it's disturbed. Now, mitochondria lose membrane potential mainly during apoptosis due to disruption of the mitochondrial inner membrane. Okay, so there is disruption of the mitochondrial inner membrane that leads to uh, loss of membrane potential. Oxidative stress or toxic agents increase reactive oxygen species, as I have told. Because what happens, suppose there is electron transport chain in a perfectly fine mitochondria that is going on. Now, once there is damage to the mitochondrial membrane, there is leakage of electrons. Now, these leaked electrons reacts with oxygen to form reactive oxygen species. Now, this reactive oxygens will make a cascade and it will keep on generating more of this. So, which damage mitochondrial membranes. Pro-apoptotic proteins such as BACs and BAC form pores in the mitochondrial membrane. This causes opening of the mitochondrial permeability transition pore, allowing ions and molecules to leak out. As a result, proton gradient across the inner mitochondrial membrane collapses, leading to complete loss of membrane potential and initiation of apoptosis. Now, one of the methods by which you can see this difference, you can actually visualize the changes in the mitochondrial membrane protein is by staining through this JC1 dye. Now, what does the JC1 dye does? So, fluorescent dyes such as JC1 are used to monitor mitochondrial integrity. A decrease in fluorescence indicates mitochondrial dysfunction caused by nanomaterial exposure. Mitochondria lose membrane potential mainly during apoptosis due to disruption. Now, how this happens? Now, suppose this JC1 polymer, when it shows red colored, when it's the portion where it shows this red staining here, it shows high film potential. But the places where it's showing green, this shows basically apoptosis because it shows low membrane potential when it stains green. The cells are cultured and treated with nanomaterial or test compound for a specific time to induce apoptosis, right? Staining with fluorescent dye, cells are incubated with a mitochondrial dye such as JC1, which accumulates in mitochondria depending on membrane potential. Now, this JC1 dye has this, I would say, inherent ability that it will accumulate in mitochondria depending on membrane potential. If incubate the cells with the dye for about 20 to 30 minutes at 37 degrees Celsius to allow staining of mitochondria. Cells are washed with buffer to remove excess dye and detection is done via fluorescence, like you can do it with in the fluorescence microscope. So when there is high membrane potential, now this high membrane potential will be in the live cells which are healthy. So it will show red fluorescence. But those cells that are undergoing apoptosis, which are under stress, there will be loss of membrane potential. It shows green fluorescence. So this red basically shows the healthy cells which has completely fine mitochondrial potential, and this green shows, uh, you know, the cells that are undergoing apoptosis. So this is how JC1 dye helps stain help gives idea about the mitochondrial potential. Now the other method for this apoptosis measurement is DAPI staining. Now DAPI binds strongly to the minor groove of DNA. We all know DAPI staining or staining. So in this case, we're talking about DAPI basically, which is used to observe nuclear morphology. Now this DAPI basically binds to cell's nucleus. Okay, if this is your spindle-shaped cancer cells and this is your nucleus, so it's basically stains the nucleus. This part, it stains the nucleus. Apoptotic cells show chromatin condensation because there is complete disturbance of cellular homeostasis in the apoptotic cells. So it shows chromatin condensation, nuclear shrinkage, and as well as fragmentation under a fluorescence microscope, which can be very clearly visualized. Like suppose this is the control. Okay, so this is control. You can see perfectly fine nucleus, well stained nucleus, right? But as soon as you start treating it with some sample, suppose this paclitaxel which is direct, you can see the nuclear fragmentation, right? You can see that the nucleus is no longer intact. Like this is the zoomed out image. If you compare with this one, I think. So this is a zoomed out image. You see the cell morphology here. This is, I guess, this is the cell, and this is sorry, this here. So there is this shows nuclear disintegration, right? Nuclear disintegration, chromatin condensation. Based on this microscopy, you can see that you can see that the nucleus is not intact. You can experimentally verify by recording these images by staining it with DAPI. So you can say that. Now let's try attempting some of the questions before we move forward to in vitro hemocompatibility tests. I'll wait for maybe a minute or two for you guys to attempt these questions, and then we'll discuss and move forward to the next topic. These questions also are from the topic that we have just now discussed. Good. Okay. Very good. So I genuinely appreciate that you guys are at least attempting and writing answers in the chat box. See, basically, these are very simple questions. Like, as I told in the earlier sessions also, like for up to the week 4 sessions, like week 1, week 2, week 3, week 4 live session, we'll basically be talking about very basic of all these topics and very basic MCQs. So in the next session, that will I think probably happen on Friday only. So in that we'll take up some of the technical questions of all the topic that we have studied in uh, in previous live sessions. But it will not only be like, okay, I'll only be talking about and just give questions, wait and wait for you to answer. No, I'll take up some of the technical questions from which it's not just, we'll discuss that, okay, this is the question, this is the answer. What exactly is the theory? What is the logic behind that question? We'll talk about some of those topics, some of uh, the aspects that demands our intellectual thinking as well. And apart from that, some topics that wasn't there in your course content but are very interesting, and especially when you're talking about this nanotechnology thing, the current applications, what are the major roadblocks, that are still existing in this nanotechnology field, that, you know, still we are taking over the counter drugs, and there is no nanoparticle based drug delivery happening at such large scale. Why the clinical transition is slow? So there are a lot of things. So we'll discuss about that majorly in that session, because up till week 4, it was more about talking and discussing and revising, revisiting the topics that were already covered in your NPTEL videos that were uploaded, and some of the extra topics maybe, and more of the basic and, kind of following an easier approach. Okay, so I think a lot of you have answered. So let's discuss it one by one. Thank you. Really appreciated that you guys are writing the question answers in the chat box. So the first question reads, which assay detects apoptosis by labeling DNA strand breaks generated during programmed cell death? So yes, the correct answer is tunnel assay only. Most of you have answered it correctly. Why? Because it labels the uh, 3' OH ends of fragmented DNA, a hallmark of apoptosis. The next question is, during early apoptosis, which cellular event is detected by annexin B assay? Now we have kind of, visually seen in the schematic also how this phosphatidylserine moiety flips to the outer leaflet. What happens during the early apoptosis, and what does annexin B stains like determines? Early apoptotic cells expose phosphatidylserine on the outer membrane, which annexin V binds to. So obviously externalization of phosphatidylserine on the cell membrane. This is what is being detected by annexin V. Which apoptotic assay measures the activity of proteolytic enzymes responsible for executing programmed cell death? Correct? Caspase activity assay. We have seen how a cascade of reactions happen and how the caspases are, activated one by one after following apoptosis. So apoptosis involves activation of enzymes such as caspase 3, which are measured in this assay. Not just caspase 3, it's not just limited to caspase 3, it varies. Now I think in this session, we'll talk a little about this in vitro hemocompatibility test, and then the next topic, which was the zebrafish model, and that thing we can cover it in the next session also. So in vitro hemocompatibility test evaluates how biomaterials or nanomaterials interact with blood components. Now, this thing is very important. Okay, so far we have seen the biocompatibility test. How do we check if the material is not causing cytotoxicity to the healthy cells, and to the cancer cells, how much, toxicity it is causing, all of those things. Now, whenever any nanoparticle or nanomaterial, if we think of the bigger picture, it will basically undergo the blood, it will come in contact with the blood. So whenever you are making any nanoparticle or any nanomaterial that is intended for biomedical applications, you know, so there would be blood contact applications, right? Blood contact. There will be blood contact. Now, one important thing is, whatever nanoparticle you have made, whatever nanomaterial you have, it should not cause, it should be hemocompatible. It should be hemocompatible. It should not cause blood clotting. It should not lead to RBC lysis. Okay. There should be no agglomeration that is happening. There should be no coagulation. So it is very important that apart from biocompatibility, there is also hemocompatibility. Your nanomaterial, your nanoparticle should be hemocompatible also. It should not cause hemolysis, clotting, or platelet activation also. because eventually platelet activation will lead to, sorry. Now the first one is hemolysis assay. So let us understand the basic principle of this. So it measures the ability of a material to rupture red blood cells, that is erythrocytes. Blood is incubated with the material and released hemoglobin is measured spectrophotometrically, right? High hemoglobin release indicates poor hemocompatibility. Now, suppose you have this, you collect this whole blood in EDTA tubes or with anticoagulant so that it doesn't, coagulate. Now, after that, you clean the RBC, like you give a wash. Now, this blood, during this blood cleaning, what you do is you collect the whole blood. You basically centrifuge it so as to collect the, RBC suspension, and then you wash this RBC suspension thrice with 1x PBS. Okay, wash it with 1x PBS, centrifuge it, and then collect the final RBC suspension. Now, once you have the cleaned RBC, you will incubate it with your nanomaterial incubation. Okay, this is RBC plus nanomaterials. So there could be two possible results. If your nanomaterial is causing hemolysis, this is how your MCT would look. But if it is not causing hemolysis, ideally it should not cause hemolysis. It will look like that. So what would happen is, see, this is the type. Now this is the positive control. So positive control is usually what happens once you have incubated with your red blood cell suspension with nanomaterials. You'll keep it undisturbed for 4 hours maybe. After 4 hours, centrifuge it again. So if there is no hemolysis, your intact RBCs will pellet down and collect as a pellet, and the supernatant will be clear. See, the supernatant is clear and this pellet is of intact RBC, right? Now, if your nanomaterial is causing hemolysis, what would happen is, suppose this is intact RBC, but once this RBC is damaged, what would happen is it will release hemoglobin, right? Now, in this case, what is happening? The ruptured RBCs have released hemoglobin, which comes out in supernatant. See, okay, that is why it looks completely colored. So you see this color, this thing. Now it releases hemoglobin and the intact RBC, the supernatant, there might be some amount, but mostly not. So after this, you put these samples in plate reader and you record the absorbance of released hemoglobin. That is why you will only take the supernatant. Without disturbing the pellet, just carefully take out the supernatant, aliquot it, this 96 well plate, and then take the reading. Record the absorbance of hemoglobin. So if your sample, if your nanoparticle has caused hemolysis, okay, if it is causing hemolysis, there will be high absorbance of hemoglobin. You will notice more high absorbance, which will correspond to more hemoglobin, and more hemoglobin means the red blood cell has undergone lysis. But if the absorbance is very low in compare to control group, what you can say that RBCs are not undergoing lysis; it is mostly intact. So, okay, so it is mostly intact. So for ideally, what is reported in literature, ideally the hemolysis percentage shall be less than 5%. Okay, it should be less than 5% for nanoparticle or nanomaterial to be considered hemocompatible. So this is the ideal, you know, range. So I hope it is clear. Positive control mostly is Triton X100 that is causing complete lysis. Negative control that is mostly taken is PBS that is not causing any lysis. And then you compare your nanoparticle, different concentration. Now this is done. Let's move on to the next method. So the next method is platelet adhesion assay. So it evaluates whether the material causes platelet attachment and activation, which can lead to thrombosis. Okay, platelets adhere to the material surface or observed using microscopy, as you can see here. If if your material is causing platelet adhesion, okay, then it's not good. So blood is centrifuged to obtain platelet-rich plasma. Material incubation, the test material is incubated with platelet-rich plasma for a fixed time. Then the material surface is gently washed with buffer to remove non-adhered platelets. Adhered platelets are fixed using glutaraldehyde or PFA. Platelet adhesion and morphology are examined using microscopy. So if there is low platelet adhesion, okay, so ideally there should be low platelet adhesion. So low platelet adhesion represents good hemocompatibility. But if there is very high platelet adhesion, see here, very high platelet adhesion, it poses a risk of blood clot formation. That's why it's written thrombosis. Okay. So this is one of the other methods that is used to check hemocompatibility. Mostly people do hemolysis assay because it is easier to do, and like you can like simply have a idea. So this is also one like platelet adhesion. You have to do the scanning electron microscopy in order to see if there is platelet adhesion happening. This is another method. Now the third one is coagulation assay. Tests such as prothrombin time and activated partial thromboplastin time determine whether the material affects blood clotting pathways. So ideally, a hemocompatible material shall not affect the blood clotting pathways. So collect blood with anticoagulant means so that when you are collecting the whole blood, in that meantime, when you're coming to your laboratory or your place of analysis, it should not coagulate at least before you start the analysis. You centrifuge it at an optimum speed in order to obtain platelet poor plasma. Okay, remember this. For coagulation assays, you need platelet poor plasma because if there is platelet already, it will lead to coagulation. Incubate the plasma with the test nanomaterial or biomaterial for a specific time at 37 degrees Celsius. Add reagents required for clotting tests, such as those used for prothrombin time or activated this thing. Calcium chloride or specific clotting reagents, then the time required for clot formation is measured using a coagulation analyzer or stopwatch. You just in this we analyze that how long or how much time it's taking for the clot formation to happen, because we have already taken the platelet poor plasma. If the clotting time is normal, we say that it has good hemocompatibility. The nanomaterial has good hemocompatibility. But if it has a very short clotting time, we call it as a procoagulant effect, which is not good. And if it is taking very long clotting time, then also it's called anticoagulant effect. So based on this is again, a different method. Now, in all these three methods, we have seen that uh, blood is centrifuged, right? It's centrifuged. So what is the difference? The optimum centrifugation speed, and in addition to that, the reason why it is being centrifuged or collected at that speed also varies. So the centrifugation condition differs because each assay requires a different blood component, right? In hemolysis we were taking red blood cells, then in one case we were taking for platelet adhesion test, then we are taking plasma. So assay, hemolysis assay, separate red blood cells from plasma. Platelet adhesion assay, that we were determining by SEM imaging. We wanted platelet-rich plasma. In coagulation assay, we wanted to remove platelets to get platelet-poor plasma. So the component of blood that is required in all these three different kind of assays are different. That is why the centrifugation speed also differs in all these three assays. So in this, the fraction used is erythrocytes, platelets, and then plasma with clotting factors. Okay. So in summary, hemolysis assay, centrifuge to isolate RBC pellet. Platelet adhesion assay, low speed centrifugation to keep the platelets in plasma. And coagulation assay, that is high speed centrifugation to remove platelets and obtain platelet poor plasma. So last method that we'll be discussing is will be based on the complement system proteins. So before coming to that, let's just very quickly briefly see what are complement system proteins are. They are a group of blood plasma proteins that help the immune system defend against pathogens such as bacteria and virus. These are blood plasma proteins which provides defense to the immune system against pathogens like bacteria and virus. They usually circulate in blood in an inactive form, and they become active only during infection or immune responses. Okay, so they are mainly produced by liver. When activated, they trigger a cascade reaction that basically enhances the immune defense during the pathogenic infection. And important proteins include C1, C3, C4, and C5. These are the common complement system proteins. These are the main functions like inflammation proteins like C3, C5 promote inflammatory responses, immune cells to destroy and all those things. So complement activation assay, what is complement activation assay? So it determines whether the material activates the complement system. Now you ideally it should not do that because which can trigger immune responses and inflammation. So ideally a nanoparticle or nanomaterial should not trigger unintended or unwanted immune responses or inflammation. So if after treatment with a nanomaterial there is presence of complement system proteins that you can identify, then it is not good. So complement proteins are measured to check whether nanoparticles activate the immune system or cause inflammatory reactions in blood. So these are four common assays that are done to check the hemocompatibility of a compound. Now, okay, okay. So I think I forgot to like keep the animation, but let's discuss. I'll just try. See, I have like kind of given the answer, but still we can discuss it. So the first question is which blood fraction is typically used in coagulation studies to analyze clotting factors without platelet inference? So we have seen that in coagulation studies we need platelet poor plasma, right? We need platelet poor plasma. Why? Because it contains clotting factors but minimal platelets, making it suitable for coagulation analysis. Otherwise, there would be interference caused by platelets. Now, in hemocompatibility testing, excessive platelet adhesion on a biomaterial surface indicates. Okay, so ideally, for a material to be hemocompatible, the platelet adhesion on the nanomaterial or biomaterial shall be minimal. But if in case there is excessive platelet adhesion, what does this shows? There is basically risk of thrombosis. So these are two questions. So it's done for today's session. Yes. Okay. So if anyone has any doubt regarding anything, you can ask in the chat box. Or maybe if you have any further doubts related to any topic, or if you want to discuss some topic of your wish in the next session that you want me to take up and discuss on that or anything, or if in future also if you need anything, you can reach out to me. The mail ID would be there, so that can be done. So thank you very much for this session. Thank you for being there for so long. Thank you. Thank you.